Okay, so I think me and Levi coordinated pretty well here because his talk is a great lead into my own. Hopefully I have a better idea how to use this remote control here thing. Okay, so really what I was charged to talk about today are two concepts. One is this outlier phenotype response and how we can use these atypical cases to bring drugs forward that otherwise would have died on the vine. And the second is this newer basket type of approach. So what my lab and what my clinical focus is to try to accelerate drug discovery, and we, as like Levi, try to use cancer genetics to do that. So just one introductory slide here. What's our approach for that? And the approach is pretty simple for those who don't think much about this. We look to define targets, and these are usually mutations that we find found within the tumor that occur somatically. We look to identify drugs that inhibit those targets, and then the real challenge for the medical oncologist is to find the patients who have particular mutations so we can give them a particular drug and see whether it works. And so the story, at least for me, began when this paper was published in 2002 in Nature by the Sanger group, showing that BRAF was mutated in human tumors. I had been working on a different target. I really looked at this paper and said this would work if we had the right drug, and I want to work on this, and my mentor was nice enough to go ahead and let me do that. And so why was this an exciting target? It was found mutated in about 8% of all cancers. The mutations really clustered within the kinase domain, and if you looked at these things in the laboratory, they were activating kinase domain mutations that were found in a non-overlapping pattern with a lot of other oncogenic alterations, which suggested that they were driver alterations. And so just to, wow, it really doesn't work. I think it's the remote, but you sure? Really does not work. I think you're out of batteries, but is there some way I can just push the button? You got another one? Oh, you got new batteries. Okay. Okay, so I'll just say next slide. That's probably a good way to do it. So just to jump forward 8-9 years, this is the RAF inhibitor. It took us a little bit of time to find the right drug. We actually went through 2 or 3 drugs that weren't the right drug, that did not work well. But when this drug came along, vemurafenib, we started to see these dramatic responses. And then next slide, this led to a dramatic improvement in survival in patients with melanoma who have BRAF mutations. So this has really been our traditional approach over the last decade, which is to identify targets, usually in retrospective studies, and then do prospective studies where we try to enrich for that target or maybe run all the patients in a particular disease. And we've seen success with this approach with some tumor types and some targets like BRAF. But there's another way, possibly a flip side, another approach, which is can we work in reverse? Can we take cases where we've seen a dramatic response, we don't know exactly why, and can we figure out why and then try to bring those drugs forward in that subpopulation of patients? And so this is the case I'm going to talk about for about 5 minutes here. This is a trial that we did in bladder cancer at Sloan Kettering of a drug called everolimus, and this is a selective inhibitor of mTOR. And like every other trial we've done in bladder cancer over the last 20 years, this was a negative study. So there were 45 patients put onto the study, and the endpoint of the study was to try to see an improvement in the mean time to progression, but as an overall average, there was no improvement compared to historical control. So the company discontinued further development of this compound. But we were very intrigued by the fact that there were several patients on the trial who did respond to the drug. And in particular, there was one patient whom we treated who's now been disease-free 3 years into treatment with metastatic bladder cancer. And in fact, she's the only one on the trial who's still alive who was enrolled onto the study. And this is not a study from 10 years ago. This is a study from 3 years ago. So a clear outlier response on that study. So these are her scans. She actually presented in the summer of 2009. She presented with metastatic disease to lymph nodes when she initially presented. They gave her chemotherapy to try to shrink the cancer down, and she partially responded to that. But then they went ahead and did surgery, and they cut out all of her sites of disease, including her bladder, and they took out these involved lymph nodes as well. And she was briefly without any evidence of disease just for a few months. But by January of 2010, unfortunately, her cancer had come back, and you can see this bulky adenopathy on this one CT slide in the retroperitoneum. And for no particular rational reason, just simply the fact that this was one of the studies we had open at the institution, she was enrolled on this phase 2 trial of everolimus. And within a few months, you can see by April 2010, she had almost a complete response here. By July 2010, she was a complete response. July 2011 was still a complete response. If I had room on the slide, I could put July 2012, still a complete response. And as of January 2013, she still is having a complete response. So we asked the question, why did this patient respond so dramatically to this inhibitor of TORC1? And so we did some targeted sequencing, the type of things we had available to us at the time, things like sequenome. We even did a whole genome array CGH to try to figure out what was going on, and all of that was unrevealing. But we had the fortunate opportunity to actually send her case for whole genome sequencing. It was actually the first whole genome sequence done at Sloan Kettering. And so we sequenced her genome. We sequenced almost 3 billion base pairs with sufficient coverage to actually call variants. And it's not that she didn't have a complex genome. She had a very complex genome. She actually has over 17,000 candidate somatic alterations in her genome. But if you look at them, you can actually now just bioinformatically get a lot of information about those alterations. The first whole genome, human genome, was only solved about 13 years ago. But now we're able to know where these mutations are, whether they're in gene coding regions or whether they're in that sort of intervening junk within genes, and potentially what their functionality is based upon computer modeling. And so if you break down where her mutations are, there's about 180 of them in actual genes. And then there were two that quickly jumped out at us very quickly. I don't know, it still doesn't work. Next slide. That we thought were the main cause of her response. So one was in a gene called TSC1, and the other was in a gene called NF2. And these were both very interesting because loss of both of these alterations, TSC1 and NF2, would actually lead to activation of mTORC1, which was the target of of the drug that she received. So just next slide. So why are these actually interesting? So TSC1 actually is tuberous sclerosis complex 1 gene, and it was actually discovered because it's found to be mutated in the germline of patients with a condition called tuberous sclerosis. And these patients get early onset frequent cancers. Oftentimes they get brain cancers. And interestingly, the same drug that our bladder cancer patient was enrolled on has actually been tested in patients with tuberous sclerosis who develop these brain cancers called SAGAs. And not only do all of those patients with those brain tumors respond, they all respond very durably. So 100% of the patients with these SAGA brain tumors who have tuberous sclerosis respond. And out through 2 years on this study that was published about 1.5 years ago in the New England Journal, only a single patient had ever progressed on the study. So our patient had a somatic mutation, meaning one she acquired through life, that was identical to the same mutation that these other patients were born with who also respond to this drug. And maybe it's not so surprising that our patient responded. So the surprise here was not the response. The surprise here was not actually the biology of the gene. The surprise here was that she had this mutation and we simply didn't know about it. So it was simply a lack of knowledge about what the underlying genetics were of her particular tumor. NF2, I don't want to go through the whole story, but a similar story as well, not quite as strong. But this was work from Filippo Giancani at our institution, published about 1 year before we made this observation, that tumors that have alterations in this gene called NF2, it's also called Merlin, another one of these, I guess, whimsical names that geneticists will come up with, that loss of Merlin or NF2 sensitizes to this same drug, everolimus, or an analog called rapamycin. So using this information, we were able to actually go back to other patients in the study. We only had tissue available on a small number of them and genotype them for alterations in TSC1, NF2, and other genes. And what was very notable is that even the patients who had minor responses to this drug, everolimus, on the study, all of those patients had TSC1 altered in her tumor. And actually, one of the things we discovered through this process is it's going to turn out probably that bladder cancer is the tumor where TSC1 alterations are most common, and we just didn't realize that going into the study. Okay, so really this is the paradigm for these outlier responders, these exceptional cases. We ran an unselected clinical trial, a phase 2 clinical trial. It was a negative study, so in the past we just would have put this one aside and moved on to the next compound. But we did see these two dramatic responders. One I spoke to you about. We figured out why she responded by doing some sort of broad sequencing effort. We've actually been studying the biology of that in the lab. And what we need to do is we need to develop a test that we can use in the clinic to find other patients with these same alterations so that we can do an iterative study where we restrict entry in that study to patients who just have this mutation. So we just have a limited amount of information. We've got 4 or 5 patients who responded with that genotype. We obviously need more to establish how often patients will respond and maybe why some of them only respond partly and others respond completely. So how do we do that? So there's really two really competing trial designs that can be used to accomplish that. And I'm just going to call the first one of these a genotyping study. And this is really, I think the best example of these is Roy Herbst's battle study here, where you genotype all patients with a particular disease, and then you allocate those patients to a particular drug based upon the profiling results. Levi mentioned this as well. And so if you have one biomarker, maybe an EGFR mutation, you get an EGFR inhibitor. Maybe if you have a BRAF mutation, you get a RAF inhibitor, et cetera. Maybe if you have no identifiable alterations that you're sure what to do with, you just give these patients chemo, and then you compare these arms to see how they did and whether the genotyping was helpful there. And so this was the schema for the battle study that Roy ran, and what they did is fresh biopsies, and then they did an adaptive randomization to give patients either erlotinib, vandetanib, erlotinib plus this inhibitor, and sorafenib. So unfortunately, there's a few problems with this approach. Unfortunately, I don't have any of the good points of the study with Roy in the audience. Maybe if I knew he was here, I'd put a couple of good points on. But just to point out the potential flaws of this approach. Unfortunately, the drugs are oftentimes not best in class, but what was available. And it must have taken Roy a long time to negotiate the ability to get all these compounds into the study. But by the time the study was up and running, there were some compounds like sorafenib, which was being used as a RAF inhibitor, which clearly were not best in class by the time the study was actually enrolling patients, especially towards the end. I don't have time to talk about it here, but I think if this adaptive randomization design is used, there's a potential possibility that actually some of the arms, randomizing patients to them, could actually become unethical or uncomfortable for the clinicians and patients as the trial goes on. And this was an issue with EGFR mutants in lung cancer because during the course of the study, it became clear that EGFR mutants were responding to erlotinib and probably needed to be given that drug and not adaptively randomized to one of the other arms. And then finally, I think this is the most important point for today. The total number of patients is generally low. So maybe you have, in this study, 150 patients. Maybe if you're doing in a nationwide consortium, 500 patients. And so it's not very efficient to find patients who have rare mutations, things that make up 1 or 2 or 5% of a specific disease cohort. Because if you have 100 patients and that mutation is only found in 1% of patients, you maybe get 1 patient on the study. And so you really can't use this design to test certain biologic hypotheses. I guess. Okay, so I'm just going to give one quote here, which is from the famous zoologist Dr. Seuss here, which is that sometimes the questions are complicated and the answers are simple. And maybe we're just asking the wrong questions for some of these clinical trials. The question really shouldn't be, what's the response rate of bladder cancer patients to everolimus? We already know this. It's low. It's low for every targeted inhibitor that we've looked at, and probably because bladder cancer is very genetically heterogeneous from patient to patient. Really, the question we should be asking is, what is the response rate of TSC1 mutant bladder cancer patients to everolimus? So how do we get to this question? How do we answer this question? So, still doesn't work, Levi? Okay, so I'm just going to introduce quickly one concept, which we're calling sort of the post-genomics discovery era here. And this is really based upon the evidence that very soon we're going to have sequenced over 10,000 whole exomes from patients with cancer. And what we're finding is that as we're doing more and more of these, we're not really discovering a whole lot of new cancer genes. We're really finding the same genes that we already knew about, maybe in different diseases that we knew they were present before, and in different combinations, different concurrent combinations. And that patients are actually increasingly having their tumor sequenced themselves. Okay, so it's, Levi laid out actually a mechanism to do this at an academic medical center. Well, in fact, this is actually happening in the community, and it's being driven actually by many patients and not, in fact, by the physicians. But these newer anti-cancer drugs are very expensive. So if a patient drives this effort, they get their tumor genotype, they find out they have an interesting mutation that could respond to a drug, they oftentimes have no access to those compounds because these drugs are $5,000 to $10,000 a month, and they're thus not available to patient off of indication. And we used to be able to, just 10, 15 years ago, we would give any cancer drug to any patient if it was FDA approved off indication, if that's what the physician thought was best. And that really changed with the introduction of cetuximab and bevacizumab, in part because these drugs were so expensive. It's not that the physician couldn't prescribe it, it's just you couldn't get reimbursed for it, and patients could not thus go on the drug for financial reasons. Also, many of the mutations that we've been finding in these sequencing efforts are prevalent in just a very low percentage of the population. And I'll show you this with HER2 And thus, we really need novel clinical trial designs to really test these biologic hypotheses as to whether a particular mutation will lead to response to a particular selective inhibitor of that target. So this is a newer type of trial design that's been bandied about. It's called a basket study. And really what you do is that you're genotyping across diseases, and maybe you're genotyping for a particular mutation. And this is just giving you the percent numbers here for BRAF. And so you're looking for BRAF across cancers. And you might find that colorectal cancer patients have a 3% incidence of BRAF, and lung cancer patients 1%, breast cancer 1%, ovarian 1%. Here's a very rare syndrome called Erdheim-Chester disease. These patients, about 50% of them have BRAF, but there's probably no more than a couple dozen in the country. And so you couldn't really run individual studies here. And you can even have an arm which is just other. Anyone else with a BRAF on some unknown arm, which would just capture any other cancers that you hadn't thought about going in. And what you do is you find that they have a BRAF mutation, and then you put them into these different baskets. And really what this is it's a parallel series of phase 2s. You're really running a phase 2 colorectal BRAF study, a phase 2 lung study, and it's more efficient than trying to open up 30 or 40 or 100 different studies. I mean, there's literally 400 different subtypes histologically of cancer that we talk about, and you really can't open up 400 BRAF V600E studies. So you can open up one basket study that potentially will bring all these patients in to answer a particular hypothesis. And we have seen that this has worked. This is actually the first patient with a BRAF V600E mutation with lung cancer treated at Sloan Kettering. You can see this is her tumor here, and you can see just 6 weeks later on the RAF inhibitor, that tumor has completely resolved. So if I was a patient and I had a BRAF mutation, and there's already a drug approved in melanoma, it's something I would like to try, and I think it's best to try these in the context of a clinical trial. And if we don't run these clinical trials and gather information as part of the clinical trials, we may find that this is just happening in the community outside of the scope of clinical studies, and we're not gathering data on response rates and gathering tumors to try to figure out why some patients respond and others don't. So what are the advantages of the approach? It allows for testing a defined biologic hypothesis. As the study is a collection of disease-specific cohorts, we can determine the effect of lineage. So one of the comments we always get is, simply because they have BRAF doesn't mean they're going to respond. I completely agree, and we're actually seeing that in the case of the BRAF inhibitor. The drug seems to work in melanoma and lung cancer, but it doesn't work in colon. But we can try to figure that out as part of this clinical trial by collecting tissue and trying to define, is that difference in response rate due to lineage-specific differences in the pattern of co-altered genes, or maybe lineage-specific stromal interactions that are causing resistance. And the key is we can actually collect tissue if we do these type of evaluations within the context of a clinical trial. Also, what's notable is that actually the individual arms can even be amended during the study. If you find out that colorectal BRAF is not responding, you can maybe amend that arm of the study after you've already proven that in 15 patients, to add on a second drug to look at a combination therapy within that context. So what are the criticisms of this approach? Well, the primary criticisms from clinicians, companies, and regulators is that you fail to identify patients who potentially could respond but lack the biomarker. I would say that's true, but I'm willing to accept that weakness in an effort to try to get at least one question answered in a definitive way. And then really the primary hurdle to do these studies, and Vince is really going to probably address this in part in his talk, is that identifying the patients remains a real challenge. And it's at least my belief, I'm standing up here so I can say whatever I want, that the screening protocol should be separated from the treatment protocol. And this is actually the most polarizing concept in this whole debate. And the reason I think that this is the case is that when we run it this way, where it doesn't matter how we found the BRAF mutation, that you're eligible for the study, we can actually utilize the entire population of 500,000 cancer patients in the country per year who have advanced disease to identify these patients, as opposed to if we do it the other way around and just have a study of 200 patients where we're screening them using a similar assay for everyone, we're just not going to be able to find enough patients for the clinical trial we want to do. So still not working? Okay. Oops. Okay. So I think one of the controversies is how do we actually define what targets would be amenable to this approach? And how do we define that something's actionable? Levi actually introduced that concept of actionability. And I'm just going to give you my thoughts on this that I put together just over the past week or two. Similar using a similar system to what we use for advanced cardio life support as to what the level of evidence is for any particular intervention. So to me, level 1 evidence for a target that would make sense for this type of approach would be that you have the same mutation and it predicts for response to an FDA-approved agent in another tumor type. So this is the example of V600E BRAF with vemurafenib, or maybe L858R EGFR and erlotinib, or ERB2 amplification with trastuzumab. But there's other alterations that have a much lower level of incidence. Actually, here's the HER2 example. This is a compilation put together by Nikki Schultz, one of the bioinformaticians at Sloan Kettering, where he just looked at 30 studies over 7,000 tumors in terms of amplification, and 48 studies over 7,000 tumors for mutation, showing that HER2 amplifications are found in many cancers, but oftentimes at low rates. And we're not capturing these patients into our studies. Similar things with mutations as well. And these mutations are scattered throughout the gene. So unlike BRAF V600E, these different mutations may actually not all be the same. And some may actually cause sensitivity or lead to sensitivity to a drug, and others may not. So just go to the next slide if you could, since it doesn't work. Okay, and then this is a patient of mine with bladder cancer as part of a retrospective study. This patient actually died of bladder cancer within 2 years of diagnosis, who has this very focal HER2 amplification on her tumor, but a patient who never saw a HER2-directed therapy simply because our system does not allow us to capture these biologic findings within patients' tumors efficiently currently to get these patients on drugs that may already exist for that particular target. So what are the other levels of evidence? I sort of would speculate or throw out there that level 2 evidence could be maybe the same mutation has been shown in a retrospective study to correlate with drug response. This would be our TSC1 example. So again, it's not, we don't have an approved biomarker drug link here, but it makes sense based upon clinical data. And then maybe a level 3 evidence would be strong preclinical data correlating with the genomic change, or maybe a non-biologically validated mutation in a gene for which level 1 or 2 evidence is already available. And that would be the case of, say, a HER2 mutation that no one's ever tried to show whether it's activating or not. But we know that that target, when activated, leads to oncogenic dependence in many tumors. So just, okay, so what is this? Actually, I just want to, one more slide. What is the study not designed to accomplish? Because I think that, again, the debate for these studies often gets held up because they're thought of as studies that are going to lead to imminent FDA approval. So what is it not designed to do? It wouldn't be designed to lead to FDA approval of the drug. It's not going to replace phase 3 studies or even definitive phase 2s in a particular disease cohort. It doesn't replace phase 1 trials. These are studies that we're probably going to be doing at the designated phase 2 dose level. It's not designed to prove that genomic profiling adds clinical value. I think we've already shown this in many cancers. I don't think it's worthwhile to keep showing that again and again, and it's a constantly moving target, because every time you get a new biomarker approved, you have to show it again in the tumors that are left. I don't think that makes a lot of sense. It's not designed to validate the companion diagnostic, and again, I think we should be separating the genotyping from these clinical trials. But as we actually could collect tissue on these, we could use it to validate in retrospective fashion the accuracy of those diagnostics that were used. And then to me, it's not appropriate for highly prevalent alterations. Things like CDKN2A deletion, if they're found in 50% of a tumor, we don't need a basket study to identify a genotype-phenotype correlation in that setting. So just next. Okay. So what's the long-term goal? It's really to define the genetic basis of every patient's cancer and then use that information to develop individualized therapies. Next slide. And so what are the hurdles? I think there's still skepticism as to the predictive value of certain preclinical data. I think this is starting to change both in industry as well as in academia. I think the major issue is delays that the genotyping is currently going to incur in terms of enrollment, and Vince will probably address this. There's clearly a lack of tissue for some patients. Even in lung cancer, where we do genotyping routinely, about 15% of patients don't have any clearly accessible tissue that you can use for profiling. There's issues of intratumoral heterogeneity. Next slide. One more. And this is an issue that we do see. This is just one example I'm going to give you, where we have a paired set of melanomas. This is the primary tumor. This is the metastatic site. Essentially, all the cells in the primary tumor have this V600K mutation. But if you look in the metastatic site, all the cells in the MET have this P10 alteration, but it's subclonal in the primary tumor. So depending upon whether you sample, you may see differences. And although this is a big concern, it's not something that would stop me from moving forward with this type of paradigm. Next slide. And then finally, we need credentialed assays, and Vince is going to talk about that. And then Levi briefly mentioned this issue of reporting and counseling. As we move to next generation sequencing, we're inevitably going to find germline alterations that predispose patients to cancer. And the question is, how much of that information has to be returned to patients, in what way, and do we need medical geneticists and counselors to be involved in this process if we do find these potential risk alleles. So next slide is just my acknowledgement slide. And so thanks again for the invitation, and I look forward to answering questions at the discussion. So thanks.